Hello, everyone. My name's Justin, and this is Martin. And today we're going to be talking about T regulatory cells and the role they play in HIV progression in humans.
So, our overview. In this presentation, we're going to be talking about HIV and its effects on the immune system, specifically with T regulatory cells and T helper cells. We're going to be showing you how, in the presence of HIV, the immune system, which normally does nothing but benefit us as humans, when HIV is present, the immune system can actually work against us. And this is going to be shown in several studies that we will be presenting to you throughout today's presentation.
So HIV was first discovered in an African village called Kinshasa, which is a region in the Democratic Republic of the Congo. And it was first found in the early 1920s, and it was observed as CIVS, which is simian immunodeficiency virus, which it was mainly infecting monkeys at the time, monkeys or apes. Okay. So what is HIV? HIV is a human immunodeficiency virus, and it is a retrovirus, which means that it will inject a host cell with its RNA and use the host cell's own mechanism of DNA replication to create more of the virus. And the initial infection comes from exchanges of bodily fluids like unprotected sex or through blood. And so there's two main types of HIV, HIV-1 and HIV-2. We're going to be focusing on HIV-1 for this report, and that is the one that generally infects humans.
So here's a general overview of how the immune system functions within the human body. We will have a pathogen, or a virus in this case, introduced to the system, and the innate immune system will identify the pathogen. This is through a macrophage. Will notice anything that is not normal, and it will present it to T cells in the thymus. These T cells will then activate, and they will become specialized to deal with whatever's present. An inflammatory response will occur to keep the infected region closed off, in a sense, from the rest of the body. And then the specialized T cells will find and terminate these infected cells. This is what normally happens, but in the case of HIV, we will show you in a bit how it actually can be complicated.
So we keep mentioning T cells. What are they? Well, T cells are made in the bone marrow, and they move to the thymus to mature, and they're able to move throughout the body through blood vessels. And T cells are the special forces or special fighters of the immune system and mainly target viruses and viral cells. All T cells have a CD3 receptor, but they're able to be identified through other receptors that they have on them. Helper T cells have a special CD4 receptor, which leads to many problems later down the line, like we'll talk about. And their main goal is to kind of jumpstart the immune system and target cells that are viral or that are infected. And they release cytokines, which can then be picked up by killer T cells, which are identified by the CD8 receptor. And they are covered in receptors and are able to bind to the virus or viral cell and kill them. Memory T cells, their main goal is to remember the virus and remember how the body fought the virus, so that if the virus ever shows its presence again in the body, the immune system knows how to fight it and can better and more efficiently attack it. And just like all systems, there's always a manager, and the manager in this case is the regulatory T cells, which can be identified through the CD25 receptor and the FOXP3 protein. And the main goal of the regulatory T cells is to regulate the environment and make sure that the reaction fits the crime, I guess. And you want this because if you have an overreaction of the immune system, this can lead to possible tissue and or organ damage.
So here's an overview of how HIV affects CD4 plus T cells, or helper T cells. The GP41 and GP120 protein complex on the exterior of the virus will need to bind to two receptors on the exterior of the host cell. The first one it binds to is CD4, and it will need to bind to another co-receptor, which can vary in different organisms. and different strain or different variations of the HIV. But when it binds to both receptors, the virus can create a conformational change. You can see it initializing in step D. It will make a break in both the virus and the host cell's membranes, which then allows for direct passage of the viral RNA into the host cell, which will then make its way to the host's nucleus and assimilate itself into the host's DNA.
So to take a closer look at T regulatory cells, again, we'll recap what they do. They're there to either promote the production of T cells or directly suppress T cells. How is this done? They're able to slow down T cells by binding a T cell to a macrophage by using the CTLA4 receptor. This will put the T cell into a dormant state, so it's not being used. They're also able to directly compete with T cells with IL-2. IL-2 is needed for maintenance and development of both T cells and T regulatory cells, and it's kind of their food source. They're also able to directly suppress T cells by releasing IL-10 and TGF-beta, which are both immunosuppressant compounds that will either slow down the cell or cause the death of the cell. Why do they do this? Like we said above, to avoid an overreaction. You don't want any tissue damage or organ damage. You want the punishment to fit the crime. Maintain homeostasis. The body likes to maintain a neutral kind of chill level, I guess, and not have massive fluctuations all around. And it's also used as a checks and balance system to make sure the body isn't spending vital resources creating T cells that aren't necessarily needed to fight off an infection.
So here we have our first study, Kitchenato et al. And their goal or their overall purpose for this study was to observe how HIV will permanently bind itself to the CTLA4 receptor on the T cell. And when it is combined with the anti-CTLA4 antibody, you can see it on the right side, the light blue antibody. When that CTLA4 cannot rebind to the B7 protein, it stays active. So this may be a good thing in a body that isn't infected with a virus like HIV, because if a cell is always active, then it's always taking out invaders or pathogens that are not natural to the body. But when the HIV binds and the T cell remains active, these T cells, which are already infected with HIV, will constantly be on, and they will constantly be reproducing and replicating this viral DNA or RNA. And this just creates a massive influx of infected cells and viral concentration within the body. So they found that when the CTLA4 receptor is blocked by HIV, T cell count went way up, and so did viral replication.
Another study done by Kleinman and his team was analyzing the role of T regulatory cells in HIV. They were the first ones to kind of switch their focus onto T regulatory cells. And T regulatory cells are resistant to expressing apoptotic gene expression when they discover viral DNA or something like that in their system. It's unknown why, but normally T cells don't have this problem. It's just T regulatory cells. So the team set out to try and figure out a way to kill off T cells themselves. And they did this by using two different drugs, Ontac and diclizumab. Ontac is made up of IL-2 and diferion toxin. The IL-2 would bind to a CD25 receptor, allowing the toxin to enter the cell. The toxin would then go to the ER, where it would block EEF2. And the purpose of EEF2 is in the ribosome, the mRNA has to move from the A site to the P site, and this is done by EEF2. So if you're blocking EEF2, you're no longer able to move the mRNA, so you're not able to create proteins, and ultimately the cell dies. So they found that this was they had a 75% success rate, meaning they were able to kill off 75% of the cells, which means, yeah, the drug worked, but they also realized too, if this was in humans, there would be serious autoimmune problems and very low counts of T regulatory cells left, so they wouldn't be able to maintain the immune system of the body, and you would see overreactions like we were trying to avoid. The second drug, diclizumab, is a monoclonal antibody, meaning it is immune cells that have been cloned from one specific parent cell. And the purpose of this was to prevent the interaction between IL-2 and the CD25 receptor. Like I said before, IL-2 is needed for maintenance and development of the cell. So it's effectively starving the cell, and the cell eventually dies. And they found a 50% success rate with this as well.
So our next study was the in vitro, or the next study we found was the in vitro study, which overall, the purpose of this study was to observe the the effects of binding dendritic cells to infected CD4 plus cells. So normally when a dendritic cell binds to a CD4 plus cell, which is caused by a T regulatory cell, the helper cell, CD4 plus, will become dormant and will not spend unnecessary resources and energy in fighting off an invasion that's not there. The purpose of creating infected cells to be dormant, or the purpose of making the infected cells dormant was to try to prevent their replication and further infection of the body. However, these researchers found that when they did this, the cells that were infected became dormant, but they didn't go anywhere and they weren't able to be infected by any medicines. So it was basically just creating a cache of infected cells that later on would be reactivated and would create a large infection in the body, a very quick infection. So they found that this process, also caused by the immune system, is another way that it can be harmful in the case of, in the presence of HIV.
So the last study we looked at was done by Cornfield and her team, and they were studying sieves in African green monkeys, and specifically the early stages of infection. They found that within the first 24 hours, there's a sharp spike in T cell count. This was followed by, around the 48-hour mark, a spike in the T regulatory cell count, which was immediately followed by an increase in IL-10 and TGF beta counts. And like we talked about before, IL-10 and TGF beta, both immunosuppressants. So basically, this means that the T cell count went up, T regulatory cell count went up. They released all this IL-10 and TGF beta, which caused the T cell count to go down. This was backed up by another study of people with HIV, and they also found this negative correlation between T regulatory cells and T cells to be true. And both teams ultimately concluded that the high levels of T regulatory cells were directly related to an increase in disease progression, because you're taking away the cells that can fight the disease. Like, yes, you are limiting the amount of cells the HIV can infect, but you're also taking away the fighters, so it's able to kind of infect freely and move around.
So in conclusion, we see that a process or a system within the human body that normally is very beneficial in keeping us healthy and safe, it can work against us. And in the case of HIV, it actually, more often than not, creates more of a replication of the virus instead of dealing with it. Our bodies are naturally unable to fight off this virus. Once we are infected with it, it does not go away. So all of these studies went to show how the system can work against us. This concludes our presentation. Thank you very much for listening.
Thank you very much.
And yeah.
Thank you.
Thank you. Thank you.